Regulation of transcription in prokaryotes. In this video, we'll explore how prokaryotes control gene transcription, speeding it up or slowing it down in response to their environment. For comparison, eukaryotes, with their many cell types and complex needs, control gene expression with high precision. Complex signaling pathways produce transcription factors and activators that turn on specific genes, sometimes just one, by helping RNA polymerase bind to the promoter and start transcription. For eukaryotes, transcription regulation is all about precise control, deciding exactly when, where, and how much of each gene is used. Prokaryotes lack that level of precision. Instead, they use broader mechanisms to activate clusters and constellations of useful genes. These mechanisms include RNA polymerase sigma factor regulation and operons. Prokaryotic RNA polymerase includes a sigma factor, an interchangeable protein that helps it bind to specific promoters. Recall that promoters are DNA sequences marking where transcription begins. Different sigma factors target different gene sets. When the environment changes, the cell degrades the old sigma factor and makes one suited to the new conditions. It's like swapping out a tool head. Each sigma factor helps RNA polymerase lock onto the right genes for the job at hand. For example, under stress, a stress-specific sigma factor is made, guiding RNA polymerase to transcribe stress response genes. The second way prokaryotes manage transcription is through operons, groups of genes transcribed as one mRNA. Thanks to having multiple start and stop codons, this single mRNA can be used to make several proteins, each starting at its own start codon. Operons have several parts. We'll focus on 5 of them today: the promoter, the operator, the activator binding site, regulatory genes, and structural genes. The promoter is where RNA polymerase binds. The operator is where repressors can bind. The activator binding site is where activators, like the cap protein that we'll discuss later, attach to help start transcription. Regulatory genes produce repressors or activators and are located outside of the operon and transcribed separately, like LAC-I. Finally, the structural genes do the actual work. They code for the proteins the cell needs. Operons are efficiently regulated through 3 key mechanisms: repress, activate, and induce. In repression, a repressor binds the operator, blocking RNA polymerase. In activation, an activator binds to the activator binding site near the promoter, helping RNA polymerase bind more efficiently. In induction, an inducer binds a repressor or activator, changing its shape to promote transcription. These mechanisms let the cell turn genes on or off depending on what's going on in its environment. Because of these regulatory mechanisms, operons are often described as either inducible, normally off but turned on, or repressible, normally on but turned off when not needed. While this covers the basics, these principles are often applied to a well-known example: the LAC operon, an inducible system. In this case, the bacterium's goal is to produce just enough enzyme to efficiently use lactose as a food source and no more. To understand how this works, we need to consider available nutrients, how they influence transcription via repressors and activators, and how gene products regulate their own production through feedback. Let's take a closer look at how the LAC operon works. The LAC operon contains 3 genes: LAC-Z, LAC-Y, and LAC-A. LAC-Z encodes beta-galactosidase, which breaks the disaccharide lactose into 2 simple sugars, glucose and galactose. LAC-Y encodes lactose permease, which transports lactose into the cell. LAC-A is part of the operon, but we won't worry about it today. These genes share a single promoter, followed by an operator where a repressor can bind to block RNA polymerase. When lactose is absent, a separate repressor gene is transcribed continuously, what we call constitutive expression. The repressor protein binds the operator, blocking transcription. No enzymes are made, since none are needed. When lactose is present, some is converted to allolactose, an inducer that binds the repressor, changes its shape, and causes it to release from the operator, allowing transcription to begin. Glucose is easier to metabolize, which is why the cell prefers it when it's available. As a result, lac transcription isn't maximized unless glucose is low. Low glucose increases cyclic AMP production, which activates CAP, a transcriptional activator. CAP then attaches to the CAP binding site upstream of the promoter, helping RNA polymerase bind more effectively and boosting lac transcription. So what has to happen for the lac operon to be fully active? Two conditions must be met. Lactose must be present to remove the repressor, and glucose must be absent to raise CAMP and activate CAP. As lactose is broken down and glucose levels rise, conditions change, reducing cyclic AMP and restoring repression. This creates a feedback loop that shuts down the operon when it's no longer needed. This system ensures that the bacterium makes lactose-digesting enzymes only when lactose is the best energy source, when it's available and glucose is not. The lac operon is a perfect example of how even simple cells make smart choices, using just the genes they need when they need them. It's efficiency at the genetic level. Thanks for watching.